The Genuine Article. Discussion and commentary based on articles from JACC.
Hello, I'm Tony DeMaria, the editor of JACC, here with Inside JACC, and here I'm talking to Dr. Fabio DeLisa, and Dr. DeLisa is a recovering cardiologist, now primarily a biochemist, professor at the University of Padua, and he's an author of a very interesting study that we're publishing that deals with myofibrillar proteins in the mitochondria and reactive oxygen species. Fabio, can you tell us about the study?
Yes, we got interested in this field through my interest in mitochondria. Mitochondria produce reactive oxygen species generating oxidative stress, so they do not only produce ATP, but also these toxic intermediates. And when the toxic intermediates rise up, there is problem for any given cell, and the link between oxidative stress and cardiac disease is well established. We know that. But the mechanism linking oxidative stress, so reactive oxygen species, with contractile failure is not so well defined. So we wonder whether the contractile machinery, the myofibrillar proteins, are attacked by reactive oxygen species. And we started studying this some 7 years ago on rat and mice, and we got evidence of that. And the evidence was that there is a linear relationship between the increase in reactive oxygen species and contractile dysfunction. The more you have these toxic intermediates, the less the heart is able to contract. And then the natural thing was to translate this concept to the clinical arena, and we could do this through the support of our colleague in Australia. So Italy and Australia are very far away, but our friendship made this thing more easy. And we collaborated, and we could demonstrate, and this will be in the article, that also in human failing hearts, there is a linear relationship between the degree of oxidation of myofibrillar proteins, especially, I'm sorry to say these names, tropomyosin and desmin, and the development of contractile failure.
Right, and you did this in specimens from heart transplants, human heart transplants, a very clever way to use that clinical setting to do your studies. Now, having shown this, does that have some implications in terms of biomarkers, potential biomarkers we could use?
I really think so. Obviously, this could be translated in analysis of biopsies, because there is no way on sections of the heart to say that there is an increased oxidation. But by performing Western blots on these specimens, you could detect that there is an increased oxidative stress, and then you know that this is related to contractile failure. But it's not only that. We are pursuing the idea that the oxidation that occurs in the cardiomyocyte reflects the oxidation that is in the circulating stream, in the circulating blood. And so we are now trying to model this in experimental animals to find if there is a relationship between oxidative processes in the blood and the oxidative processes in the heart. If this is for real, then we are in good shape, because then measuring the oxidative stress in blood, we will know that the heart is suffering.
Yes, and it would give us some potential therapeutic targets in understanding this pathway. I think it could be very valuable. These insights that come from basic science ultimately provide us with our most important clinical tools. Thanks very much for being with us. For Inside JACC, I'm Tony DeMaria.